is a very nice field, very good field. If you are really interested in clinical research part of, very good with the laboratory data and all this thing. Okay, we have so many different roles in the clinical research itself. Okay, starting from clinical coordinators, monitors, the our doctors are there, investigators are there, auditors are there. Okay, so many different roles are there in the industry where you can play one of the role and excel yourself. Of course, there are so many things which we need to keep in mind. So I will take you through all those things. What are what are the actual, what are those things? What is actually clinical research? What are the documents which we are dealing with? What are the different phases of clinical research? All these things we will try to learn in this coming few sessions. Okay. So the class is going to be almost for 3 hours. However, if it is permissible, I will if it is okay with you, I'll take a break about 15 minutes or 30 minutes. And the session is going to be extended in the similar manner only. Instead of stretching directly to 3 hours, we can take a quick break so that we all can have our whatever the breakfast, lunch or brunch, so that we can have energy and continue. Okay. Sorry, anybody who is not on mute, please go on mute. So, and in between, we'll take, we'll go for 1 or 2 minutes of bio break also. So, not to worry about that part too. Okay, so Sana, I kept you on mute, maybe some disturbance. Okay, so this is a big group, guys. But again, feel free to ask a question if you have any. Sana, you have any issue with the mic, I think. So I'm just trying to keep you on mute and I think it is getting unmuted again. Please keep yourself on mute. It is again getting unmuted. Sana, if you are audible, please keep yourself on mute. Sana Sugwakar, can you please keep yourself on mute? I'm trying my best to keep you on mute, but it is getting unmuted again and again. Sorry, guys, I'm not able to take up from here because of Sana. Right now, I'm just trying myself best. That's the problem with the sometimes the big group. Okay, fine. Hope that not this time. She will keep on unmute. Okay, so I'll start my session with the first 2 questions which I'm putting in front of you, and I'll expect some good answers. First, how long do you think that it takes for a new drug to get approved in the US or EU or anywhere in the world? Okay, how long do you think that this drug discovery and development cycle takes? And how much investment do you think it is required to bring a new drug into the market in terms of monetary or money? If you can tell me in US dollars, international currency, approximately 100,000 or 1 million or 1 billion or something like that. Okay, so just tell me whatever the good idea you have about these 2 things. Okay, 10 to 12 years, 10 to 12 years, 1 billion USD. Okay. So whoever has already seen my earlier previous slides, okay, maybe you're answering it very much correctly. That's one of the issue actually I'm facing sometimes, that my other slides actually goes, or my presentation goes earlier to the students. 8 to 10 years, $1 billion, 10 to 12 years. Okay, fine, great. So you all are actually very near to the answer. So basically, it takes about 12 to 15 years roughly. And also, if we calculate as per our professional career, it is roughly around 1/3 duration of our entire professional career. as you can see that the professional career is going to be like, we are expecting is 30 to 40 years, depends how early we start our work and how long we can go. Okay, if we divide that entire career in the clinical research itself, we can see almost 3 to 4 drugs, okay, to come up into the market in this entire career, because almost this takes 12 to 15 years on an average. Not talking about COVID-19 vaccines or drugs, which these are some emergency use type of vaccines or drugs which we need because of the matter of time. We just need right away these things, and that's why everything is actually in the expedited mode. This is not the regular clinical research. It takes almost 12 to 15 years. Anywhere in the any part of the world you go, this is the in general time which is going to be required for entire drug discovery and development cycle. Okay, $1 billion, that is an approximate money part, or you can say investment we need to do in taking out a drug. Again, this is not a fixed amount, it is a on an average or approximate amount. Okay, it can be plus or minus to this figure, but in general, if I will say, okay, it is still 665 plus employees in US. If they earn $100,000 salary, that is around one. I'm really getting disturbed. So whoever is CR@21 Redmi Note 9 Pro, please go on mute. I'm trying myself to keep on mute. So a lot of you are actually not logged in with your name. I request you to make sure that you either rewrite your name so that we can connect easily. Again, that's the same problem I face with Sana with CR21 Redmi. Okay, fine. So $1 billion is like an approximate amount. Okay, 665 plus employees. If we give their salary with $100,000 per year for 15 years, then we will match this big figure. So you can see that it is actually not a small investment. It is really, really a big investment which we are putting through over here. Okay, really big investment, and really so much time investment also we are doing. So this clinical study is basically not an easy project. It is basically entire drug development is a massive undertaking in terms of time, in terms of effort, in terms of monetary part. It is really, really a massive undertaking. Hence, every single sponsor, every single company, they take a lot of risk. Basically a very calculative risk also, okay, to take this particular product, drug and development. And it is not a guarantee that your product is going to be approved only after 12 to 15 years. There can be a possibility that your drug do not show particular that benefits, or it may not show that benefit risk balance when authority checks. Maybe there's a possibility that it cannot show you that much good benefit against the risk. In those conditions, you may got this thing, that is your products are going to be rejected. It hardly happens 1% of the drug, but it can happen. You cannot decline on that part. Okay, now there comes the another thing, that here we have the selecting the right partners. That is another important thing. Okay, who prioritize sync with you. So nowadays we know that this research or taking out the drug, it is not one company task usually. Okay, many times we have seen that more than one company, they collaborate together. One is putting up money, one is going to do the marketing, so they do a partnership. So we need the right partners also in doing this research, okay, who have a positive interest in this part. Focus on high quality and fast turnaround is another important part. Okay, we have seen, especially in this COVID-19 also, nowadays we actually, as a human nature, we have not much patience. We need the drug ASAP, as soon as possible, right? Because of which it is not the time which was there 30 years ago or 40 years ago, when we used to do a slowly in a particular manner. We are doing a research and taking out a product up to so many years like that. Nowadays, this entire drug development cycle, we are trying to squeeze it, okay, so that the drug can come up faster into the market. So quick turnaround time, plus the high quality. So high quality, I hope you have already seen so many times in the news, when it started coming, that Pfizer vaccine showing 95% of efficacy, another vaccine is showing 80%, another is showing 92%. They are competing for the efficacy percentage. Why? Because people are no more dumb. Okay. Nowadays we have internet and everything to check that which drug, which vaccine is the best one. And as a human nature, we always want a high quality stuff. Hence, there is a lot of focus from industry side nowadays on the high quality and the fast turnaround. Plus, it goes a long way in avoiding the missteps in moving your drug program to success. It's a really long project. Okay, from the day when you start thinking about a target, and then taking out a molecule, then taking out a particular lead molecule, then going to the animal studies, then to the human studies, phase 1, phase 2, phase 3. It's a very long journey, and we need to avoid any missteps. Any single misstep can be harmful for the entire project. So, hence, all in all, this is just to say that let's think that there is a huge, massive undertaking in the clinical research. We are dealing with the public life. Hence, this is very critical to, to put your entire effort in taking out the drug from all the side, considering all the guidelines. Now, coming to this point, the next one, why are the new drugs needed? I'm just putting this question also in your bucket before I answer. So, what do you think that, whatever the reason it comes to your mind, okay, you can just write down. What do you think that why we keep needing the new drugs continuously? Why we are not stopping here itself and says that we don't need any more drug in future? Pharma industry also can stop and says that, okay, fine, no more drugs we want to create in future. That's all. Whatever we have is enough to take care of the human beings or human lives, everything. Why we are still going on with the research and research and research so much, okay? Or every year we are coming with so many drugs. Okay. New disease, resistance mechanism and organism, new virus, side effects of the drugs to find efficient and potential molecule of a drug. Very good, very good. Low efficacy of the drug. Correct. Anything else? Any idea related to, right, patent expiry? That is correct. Any idea related to the towards the industry? Any thinking from the industry perspective? Okay, fine. Great. So you all contributed a lot in answering the question part. Okay, I think yes, industrial activity. It's basically to sustain the industrial activity to find a drug which have multiple actions. Correct, correct, correct. Okay, that is correct. But actually, for the last part, for multiple actions, I would like to say actually we are not looking for multiple actions in one go. Usually, we are targeting for a certain indication in clinical research, and in future, with the help of pharmacovigilance department, when we come to know about the other side effects and other things, okay, that can be taken care to go for another new indication research. But usually when we run the research, we go for one or two indications, with that only that these drugs can be effective in this 1, 2 or 3 indications, some targeted indications, but not like very 5, 6, 7 indications. Okay, usually we are not taking out that way. Okay, so the answer is basically we can take out our entire thing in these main buckets: unmet medical needs, cost of therapy, and sustained industrial activity. Okay, so these are the important buckets in which I can provide my answer. The first one is a new disease, low efficacy, side effects. Okay, that means the new disease are going to come on and on. Okay, we cannot restrict them. Even our antibiotics and other antimicrobial substance which is getting resistant, so we can say that the new strains of our bacteria, microbes, they are going to come on again and again. Similar like this COVID-19, right? We try to tackle some of the strains. Now there's another strain come up like a Delta version or something like that. And who knows that in future there is going to become up the new one. But definitely it is a for sure there is going to be the new one also. As the common cold virus, every year they are actually, it is changing itself a lot. Okay, so the microorganism strain will come up. We will have a resistance in the particular antibiotic. Similar way, we will have the new other disease which will come up like SARS, Ebola, etc., XYZ things. Okay, on for which we need the drugs. We cannot stop creating the drugs first. Secondly, the low efficacy and the side effect part. So we cannot say that this the first generation of the drug which we have, that's enough. Okay, we cannot say that because we need the another generation of the drugs which have the low efficacy, which have the high efficacy compared to the old generation. Plus also we are targeting the side effect. We have seen that the first generation usually have the more side effect, another generation have the less side effect and more efficacy. The new generation will come up that have more and more potential effect and less side effect. You can compare a simple example of the anti-allergic drugs. Okay, the first generation of anti-allergic drugs has so much drowsiness, sleepiness, and all this type of side effect. Okay, with not much effect on your sinus, cold, cough, allergic part. And the latest generation of the drugs, you can see that those drugs actually have almost zero drowsiness effect. You can take in the daytime, go for your work or study, and also you get the relief within 15-20 minutes with your runny nose and the cold and cough. So these are like latest generation of the anti-allergic drugs or antihistamines. So this is why we keep on researching for new drugs. Another important answer is cost of therapy. See, for the long-term disease like the cancer, like diabetes, okay, blood pressure, or any other drug, any other disease which are long-term disease on which we need to take the drug for long time, right? For those particular disease, okay, we need to continue keeping our money. We need to continually spend the money for them. Okay, this is directly affecting our every month or so the budget. So we need to keep a tab on the cost of therapy. That is very important. If you have anybody in your family as a diabetic or blood pressure person or cancer person, who having the cancer, definitely you always see that which drug the person is taking. Is there any alternative available right now? Okay, nowadays so many this online pharma websites are available like PharmEasy and all this NetMeds, etc., where we can compare, right, the drugs. Okay, so I'm sorry, whoever is raising hands, I will request you to put your question on the chat box. I will take up from there. So from here, nowadays we can easily compare even the single active ingredient compounds also, that which one is cheaper, okay, or from the good company, so that we can continually taking that brand. Why we are doing? We are keeping a tab on the cost of therapy. Hence, same way the company also thinks that the cost of therapy is so important that they need to keep an eye on it. Okay, they need to research on some alternatives of the costly medication which are available in the market, so that the general public also have the another options. Maybe not the better drug than the one available in the market, but at least better in the form of price cost. Hence, this is one of the another reason to have some other alternatives of the existing drugs in the market. Another reason is the sustained industrial activity. Of course, this is very important if you think from the perspective of industry, to especially the brand companies, the brand product companies like this Pfizer, GSK, and the Novartis, Eli Lilly, all these big companies, Merck, okay, which have the brand products in the market. For them to survive, they need to come up with the new and new patents as soon as possible, because from them on that only they can earn a good amount of profit or money, so that they can sustain their industrial activity, they can pay for their employees, and they can keep the industry running, because of which they need to keep on researching, keep on taking out the new and new products into the market. Hence, in general, wherever, whatever picture you want to see from these three, okay, either from the general public view, that is unmet medical needs or cost of therapy, or from the company view, that is sustainable industrial activity, we need the new drugs to keep coming into the market. Hence, the clinical research is again a non-ending domain. It will automatically always keeps going on. Okay. So sorry, somebody was raising hand. If there is any question, please let me know. I'm just keep checking the this chat box. If you have any, please let me know on the chat box. Okay. This is example of the some costly drugs in the market. Okay. You can see that these are the drugs which are available in the market, very costly drugs, from the top 20 costly drugs as per the January parameter when I actually completed this slide. Maybe there is a change in the sequence now, I'm not sure, but if you even go with this particular sequence, you can see the myalept over here on the top, the first one, you can see that it cost you $71,306 in a month, not in a year, I'm talking in a month. It is like an injection every week or so, we need to get 2 or 3 injections of $7,000, something like that, every single injection. For some a rare disease it is there, but still you can see the cost per month. I don't think so that any normal or individual medium class person can afford this much. So for these type of drugs, even you go on the 20th drug, you will find $27,300 for this Delga, this particular product, for every month. This is again a huge amount, right? So for to take this thing, to afford this medication, we need to have so much money, right? Otherwise we need to look for alternatives. Hence the other companies, they also think in the same way to provide the alternatives with this costly medication. Now, any question anybody have in the chat box? Okay, so let me take you to the next slide. So now, since we have a basic idea that why we are heading towards clinical research, what are the opportunities we have in the clinical research because of this continuous evolution of the drugs, new drugs. It's good time. I'll take you to the drug life cycle. Okay, maybe you want some of you are aware about the drug life cycle earlier, but please bear with me, or you can brush up your knowledge part. This is very important, okay, because this can also be asked in future. So the drug life cycle actually starts somewhere from the drug discovery or research and discovery part. Here we do the discovery of the new drugs. Then it comes, once we have the particular lead compound or the drug candidate, then it will go to the development stage. In the pre-development stage, that is called as preclinical study. Animal testing is going to be there, okay, to understand the drug safety and the efficacy part also. Then after the preclinical, we are going to file the IND application, investigational new drug application. After that, if the drug application is going to be approved, then you can start using the product into the humans. It is the second development stage. Okay, important part after preclinical for the human trials or clinical trials. Once we are done with the phase 1, phase 2, phase 3 of the clinical trial, then you can take this drug to the this entire experience as in the form of your application. It can be the ND application, BLA application, or other applications. You can take it to the particular review stage for the authority. So in the authority will check your file if everything is fine or not. Okay, if everything is good, you will get the approval to market your product. And the cycle goes on during the post-marketing. The another thing starts. Okay, it is also called as clinical trial phase 4. Okay, that is post-marketing surveillance. We keep an eye on the drug for its side effects. Okay, and for some other factors. So pharmacogenics is one of the PMS study which we do once a drug comes into the market. So that is again another domain which I am expertise I have in it, pharmacovigilance. So I will let you know about PV a little bit in some other class, somewhere in between, or by the end of the session I will give you a little bit brief about the PV also. And during that entire thing, either the cycle stops here, that means the PV or the post-marketing activity just goes on, or there can be the other thing that you may find some another side effect which can be a potential indication for the drug, and you may take it to the new research again, that is again to the preclinical level, and say that okay, this particular candidate can show one more effect. Okay, let's say minoxidil usually we used to use for the blood pressure related medication, but it also shows the hair growth as a side effect. Now this can be an indication, so let me take it for the new next again research this same product minoxidil, and let me see that if it if I can take it as a new brand product with the new indication of hair growth. And if everything is going to be successful with the same cycle, you may end up with another new product with the same active ingredient. So the cycle can repeat again. Okay, so this is how the drug cycle life cycle goes on. If you don't have any such new indication or other thing, not to worry, the cycle is not going to be closed loop. This is you can consider as an open loop, and then PV activity will go on. Okay, so PV is like not an operation, not a project, it is an operation, it goes on and on. So this is a general about drug life cycle. Now broad steps in creation of the new drug. Now whatever I have just mentioned, the same steps are here too. There is a drug discovery research. We are going to identify the potential therapeutic target. Okay, so what is the particular target is there? We are going to get a particular lead molecule from that target also during our drug discovery. Then we go to the another phase of testing into the animals. Okay, if that's successful, then we are going for the testing into the humans. Okay, so allow me 1 second. I think there is some mismatch. The other thing, so the third box actually need to come earlier, and the second comes after that, in the next third place. So there is some mismatch. Preclinical actually comes earlier. Then comes the clinical trial, and the clinical trial through approval of the drug by regulatory authorities. So drug review is going to be done by the regulatory authorities. They will check your file, validate if everything is correct or not. If everything goes fine, you can commercialize your product. So it is like marketing of the product sales as per therapeutic applications. Okay, so this is the entire cycle. So we can just see one more time the same thing. Any doubt, any question anybody have? This is another important part. Screening. This is another important part. That is the entire all the phases whatever we have, discovery development part. We can just little bit see in depth. So basically the first one is the drug discovery, where we are going to do the target identification. We are going to identify that what disease we are targeting. Okay. And then we are going to create some compound or find out, discover some compound which can work on those targets. Once we have that, then we will move for the preclinical study where we do the testing on animals, cellular disease models, animal models. Okay, all this thing we are going to do. After that, there is going to be the IND application. We are going to file the investigational new drug application. Okay, company submits this to the regulatory authority. In USA, it can be FDA. So FDA authority for USA, it evaluates the application. For India, it's a different authority. For European region, it's a different authority. Once this get evaluated and everything is fine from the animal studies, you will get the permission to run the clinical trials, that is human trials. In human trials, we have various phases: phase 1, phase 2, phase 3. Phase 1 is like for healthy volunteers, where we are mainly focusing on safety part in a small group. Then comes the phase 2, which is on the diseased individuals. Okay, so that we can have not only safety but efficacy parameters. We have more data. Phase 3 is another important phase where we have the multi-site. Okay, we have the multi-different geographical site clinical trials going on. So a big number of patients actually get included into this phase. Okay, of course, diseased individuals, so that we can have a big data as an evidence to the authority that the drug is working fine or not. Since we have a good data from phase 2 about the safety and efficacy, hence in the phase 3 we can see the same effect in the large population or in the big size, sample size. Now we will go a little bit into depth in all these different phases one by one to understand. Drug discovery and development timeline. So here we can see this picture in the terms of compounds, like how much we have in the beginning, and then slowly it gets down, and also in the terms of time, that how much each phase takes the time. Almost 10,000 molecules or so we have in the beginning. It get drilled down to almost 250 molecules, or 50 molecules, 50 to 250 molecules in the preclinical stage. Okay, with the drug discovery, we have so many different tests and screening methods, because of which this 10,000, 15,000 compounds get screened out, and we are going to left with the 50 to 250 potential candidates, which we are going to take it to the preclinical level in animal testing. During the animal testing, we are going to check that how many products are so much safer and effective. So you can say this is another screening in the animals. With the help of this, we will come out with the final 1 to 5, or it can be more also, but not that much. Usually 1 compound or up to maximum 5, we usually are going to file the IND application, that we want to try these many products into the human trials. So once you got the approval, you can run the clinical trial, and almost this 1 to 5 compounds you will test into the human. And then after that, you will finally going to be left with hardly 1 compound or a maximum of 2 compounds, which are actually showing the good efficacy and the less side effect. Okay, this is actually showing this is actually to tell you that this is the best compound I have for the review purpose. Hence, after the review, maybe you'll end up with final 1 compound for which you get the approval. That is going to be your final product. So this is the entire cycle. If I see this entire cycle in the terms of time, it takes almost 6.5 years from discovery to preclinical stage, and then almost 7 years to complete this clinical trial, the same time which together drug discovery and preclinical took. It's the longest phase, clinical trial. Okay, then again, review phase is also not small. It takes 6 months to 1.5 years. It can be more, depends on how well the review is going on, in-depth review. Okay. After that, your product can be marketed. So this is the entire thing in terms of molecules, compounds, and the years. How much years it takes for every single stage. All right. This is just in general a picture to show you that a particular cycle of our entire drug discovery and development. Okay. Allow me a minute. So this is basically to show you that initially we have a specific idea. After that idea, like, that means what we have in our mind that we need to target the COVID-19. Like this, there is an idea. Our project team makes an explanatory development statement that we want to go with this particular this project or not. With the help of that, we started creating the compound, and then finally screening them with the help of computers and robotics technologies, lot of high throughput screening technologies. Okay, with the help of this, we will finally come up with some compounds, and we are going to do the test on animals. Okay, with the after animal studies, when we have some good data, we actually going to test in the healthy volunteers in stage 1. And then when comes the stage 2, where we have 100 to 300 patients, diseased volunteers. With good data, we can go for the multicentric trials. In the different centers, it can range to 3,000 to 10,000 individuals, or maybe more, depend on the project complexity and the project budget. Okay, with the help of all this thing, we get a good data, and then our CDM team and other teams, they will analyze this data, okay, and create a report. Finally, this report get reviewed, and we get a final new medication into the market. So this is the entire long cycle of the drug development and research. Any doubt anybody have till now? Okay. Drug discovery part. So here we have few steps. Okay, we can see that now I'm just going a little bit in depth into the drug discovery. So what is drug discovery? Basically, we are targeting a particular target. Okay, now before I go ahead, let me ask you this question. What do you mean by target? Any general idea anybody have? Okay, any example of the target? You can write down on the chat box. Okay, antigen. Any other targets? So correct, some of you have written it correctly. Receptors, tumor part, fine. Tumor is like a particular cell of the body only, and particular receptor, organ specific target, that is correct. What else we have? Specific drug molecule. No, drug molecule is not our target. Our target is our body part, basically. Gene, The virulent part of virus, that is correct, but usually that is correct. Yes, like some of our antivirus drugs, they are also directly targeting that part also. Infectious agent, antigen, fine. Okay, so mainly the target is the particular body part. If I just say directly, a body part. Okay, or if it is a virus or other thing, maybe that also we are targeting a certain a certain protein or receptors or like that also. In general, if I say, you can just consider in most of the medications we are targeting either one of our cell or receptor, enzyme, hormone. Okay, all these things, these are our targets. On which particular target what we need to work on, okay, so that we can have a particular target confirmation validation we have. So let's say, let's just pick up one drug. There is a particular blood pressure medication. Okay, a calcium channel blocker is there. So whatever our cell calcium channel is there, we are going to block that part. Okay, for a particular cell, our from the related to the cardiac cell, we are working on that part to create that particular amlodipine type of drug. So that why we got our target now. So now we need to validate the target. We cannot just go as a hypothesis and say that this is my target and that's all, I just want to make a drug. No, we need to validate with the help of different tests and other technologies. Okay, we need to validate this target. Hence, after the validation only we can say that this is my final target. So that is the target validation part. Then comes the assay development. Assay means test. So on the other side, once we got the target and everything is there, on two sides our team starts working. One side they are started making the creating the test. Okay, these are on the computers, robotic technologies, laboratory, our chemical test and other things also. Other side, the teams are also started screening or actually finding the compounds, those 10,000, 15,000 compounds which can attach on that particular target site and give some effect. I'm not saying the potential effect right now, which can give some good effect. Okay, so this is two sides we are working on. After that, what we do is we are going to take these compounds, whatever we have identified, 10,000, 15,000 compound, and we are going to run through the high throughput screening. High throughput screening is nothing else. It is basically the computer test or robotic type of technologies, liquid handling technologies, which are nowadays fast technologies. We have to screen the thousands of compounds with so many tests in quick time. So millions of tests can be performed in a quick time to screen the compound. So we are going to screen this compound and take it down to certain numbers. Okay. Once we have those compounds in our hand, these are called as hits. And after the hits, we are going to go for the lead confirmation or to finding the leads. Now, what is lead? Lead are the very final compound, okay, which are going to be done after the another level of screening of the hits. So you can say like out of 10,000, 15,000, we finally come up with the 5,000 hits, or let's say 2,000 hits. We cannot test 2,000 product on animal. We want further filtered products which are very much showing potential and the less side effects. So those are lead compounds. So we will go for another level of screening to get the lead products. So lead optimization is going to be done. Finally, after this lead identification, we are going to do the optimization. Now, what is optimization? I hope you all have read earlier about the this optical isomerism, where we are checking the levo and dextro rotatory part. Also, we have seen the medicinal chemistry, or in our 11th 12th chemistry also, how we are dropping one atom and adding some another atom, something like that, to change the structural part of a particular molecular compound. So this is what we do over here with the lead. We try to optimize this product to get the desired product. This is called as lead optimization. Here we are we are just trying our best to whatever the final compound leads we have to make it more and more better, so that whatever the other side effects we are expecting, that can be reduced more and the potency can be increased. Till now, you can say over here the drug discovery actually stops because we have the final drug candidates in our hand. Biological testing in this picture actually showing you the preclinical testing only. Okay, it is just to show you that the cycle can be repeated in case if it is preclinical testing we are doing and you find that this candidate is good. Okay, it is giving me very desired side effect, but there is one desired effect, but there is one side effect or an adverse event is there along with it. So can I remove this adverse event or side effect after dropping or adding some molecule or doing some molecular changes? If yes, then you will go back one step and optimize this lead compound and try again in the animals. So this cycle can be repeated because here you have not yet filed any IND application also. Okay, so till the lead after the lead optimization, when we have drug candidates, our cycle actually stops. Okay, from the drug discovery part. This is what I'm just telling you everything. So I'll just give you 2-3 minutes to do a quick self read. Okay, so that you can understand whatever I have just mentioned. Okay. So I hope you all went through the entire thing I just mentioned. Okay, this is the same thing I explained over here. So there is a target identification and validation part, then there is a assay development and screening part, then there is a high throughput screening part, then there is a hit to lead, and finally the lead optimization. Okay, so this is for your reading purpose. Whatever I have just explained, it is all written in the same way. Okay, you can see all the steps one by one mentioned over here. Okay, there is a question or something. Let me see. Target validation means what? So, Sumit, target validation is basically when you are validating a particular target, analyzing, that is putting some evidence on it. Okay. See, you cannot just say that my goal is to get into the clinical trial industry. Okay. Reason being that every goal, everything, every such target, but you need to validate that. Are you sure you want to go in clinical trial industry? How you validate? You are going to first understand this thing via training or via your self reading, that what is clinical research. Okay, what is the scope in the clinical research? What is your future in the clinical research? Okay, how interesting is this field for you? Okay, then you are going to have some good examples in front of you from the industry, from your near dear ones, that okay, they are doing good, earning a good amount, and also having a good future. So many things you collect in your mind, and finally You decide that yes, now you have to go for your future. Your future is going to be there in the clinical trial industry. So that is going to be one of your important goals. Okay, so this is how you are going to validate. You have your target, you are going to validate the target. Same way here in industry also, when we have a particular target, a gene or a particular protein molecule on which we need to work on to eliminate this disease, we are going to validate the target. Okay, that this is my target. Here I'm going to work on. Okay, and this is how I have to this is my target because of these many tests. I can see that this is the only particular target which is showing this particular result for the disease. So somehow with multiple tests, multiple things, you need to validate the target, whatever you have identified earlier. Okay. Can you explain the hit to lead process once again, Adeline? Yes, I can provide, no issues. So hit to lead is like a second level of refining. I'll again give you a general example to understand this process better. Let's say in an exam, okay, right? Like in our UPC, or if I'm correct, that IS exam and all this exam, we have two levels of test, right? The main exam and then the another exam is there. So two stage of tests are there, and then following with the interview, if I'm correct. So with why they are doing all this thing, because out of lakhs of students who are appearing for this government exam, right, they want a selective few individuals who are really have the potential to run the country at that level. Okay, to run the particular that particular district or whatever the things as a collector. So hence they start from the level 1, that is the first screening or first exam, then they go for the another exam, that is the second stage, to filter down furthermore, and then they may have only hardly 1,000 individuals or so finally to get interviewed. That is the third stage of screening, and finally they got those 100, 200, whatever the individuals they are acquiring every year. Those are like our final IS officers or IPS officers, right? So that's the same thing we are actually having. Thousands of compounds in the beginning. First level of screening will give us the hits. Those compounds, actually we call them hits. And now somebody asks you that, fine, are you done with hits? You say yes. Okay, fine. Now go with the second screening so that we can have leads. So then there is another checklist, the tough checklist, to take out those products also through that process, so that we can have the leads, the very fine defined particular products or compounds, okay, which can be taken to the preclinical study. Those are called leads or the drug candidates. Okay, so if I try to I'm just trying to attach with some outer example. I hope that is good to understand. So that's how we are going to have the leads. Everything is going to be the particular test only. Okay, these are mainly computer-based tests. Okay? Liquid handling devices, robotics technologies, also the test in laboratories. Okay, so with the help of these assays, these tests, we actually find out these molecules. What is the purpose of hit to lead optimization? The purpose is same, Pratiksha, that we want the final product which we have. Okay, hit to lead is not optimization. Hit to lead is a process, and lead optimization is actually another process. So lead optimization, I think hit to lead I have just explained as per Adeli. Lead optimization is the another process in which we are actually optimizing our product. That means whatever. So see, now I'm just taking you to the next level in the same example of that our officers. Okay, so you have the officers now with you. Are they ready to go and serve the country right away after selection? No, right? They are going with the training. Why? Because we want them to further refine themselves to get those skills. Same thing, or if they have some extra skills which are actually not required for the job, they want that behavior to be eliminated. Same thing we do with our drug candidates. We have this lead compounds in hand, we need to optimize them. We need to change them a little bit in such a way, we manipulate them in such a way that it can give the more potency and the less side effect. Okay, So those things we can do by eliminating either one or the other particular atom or a molecule out of this compound, or by doing this optical isomerism type of things. So medicinal chemistry basically plays a lot of role over here. Good. Sumit, Adeline, Pratiksha, do you got your answer? Great. Great. Thank you. So let me take you to the next particular question part. So here the drug discovery process ends when one lead compound is found for a drug candidate and the process of drug development starts. At least one lead compound or drug candidate should be there to say that this process is finished. At least one. It can definitely, we usually come up with 50 to 250 compounds by this stage, but at least one should be there to say that now this process is finished and we can move to the preclinical stage. Okay. So the preclinical stage, as you have seen the animal, we all know that it is basically an animal testing stage. Okay. Till now, we have not yet started clinical trials. So I will take you to the clinical trials for which you are all here and you have interest. But these studies are also important because especially when you're going for preclinical trials, when you're in the interview, you may have some questions which is related to the preclinical or discovery part. That can also be there. Okay, especially for the beginners in the clinical trial job. So preclinical studies, basically, if I just talk in general, these are the for non-human subjects. Okay, for efficacy, toxicity information, safety information, pharmacokinetic information. What is pharmacokinetic? Anybody from the pharma, participants? Can you explain pharmacokinetics on the chat box, or the medical people, or whoever has the idea about pharmacokinetics? No? Okay, Ishra answered. Effects of drug on body. What drug does to body? Okay. Anybody else? Come on, guys. I think we have so many from the pharma individuals. Study of drug absorption. Great. Anybody else? Absorption, distribution in body. Correct. Fat determination, study of drug absorption, distribution, metabolism in body. Okay, Vishal. Good. Metabolism and excretion. Yes, Nikita. Great. So whoever answered, you all answered correctly. Okay. Maybe some answered in pieces, some answered correct, as a complete. So basically, so the other individuals over here, like from biotech and other background, if you have not much idea about pharmacokinetics, I would like to tell you, please don't worry. It is not about all the pharma individuals who can work on because they know the pharma term better. It's not like that. There are only few terms which are basically related to the specifically to the pharma or medical industry. It's as general. If you have the life science background, you can work around, so no issues with that part. If I just say, it is basically one second, one of the answer is enhancing efficacy and decreasing toxicity of a patient. No, not really. Okay, not really. So first two answers, first two individuals, I think Ishra and somebody wrote that what drug does to the body, if I'm correct. So that is actually not the pharmacokinetics. Sorry to say that. Answer is that is actually pharmacodynamics. Okay, pharmacokinetics is basically what the body do to the drug. Okay, that means when the drug comes, how the body is going to absorb that drug, absorption. Okay, how the drug is going to distribute in the body in various organs. Okay, finally how it is getting metabolized and excreted from the body. So that is all comes under the pharmacokinetics. So when we start studying about the drugs absorption, distribution, metabolism, and excretion, what we are doing, we are actually doing the study on the drugs pharmacokinetics. Okay, so whoever have no understanding, I hope now you got a good understanding. That's all you need to unlearn about the pharmacokinetics. Don't go much in depth right now. Okay, it's yes, it is a quite a separate chapter in pharma if you are going for pharma. Myself, I'm actually an M.Pharm individual. Okay, MS actually from the US. So and apart from that, I also have the postgraduate diploma in clinical research and PV pharmacovigilance. So hence the pharma terms, definitely I'm very much aware about it, but I'm again saying that individuals who are not from the pharma background, it is really not that much important that you need to go with the pharma thing to get into the clinical research or in the pharmacovigilance type of departments. Okay, you need to have some information about this, but main information about itself, the clinical part. All right, so we have these things. We actually need this efficacy, toxicity, pharmacokinetic part of informations from this entire study. Okay, scientists mainly do all the tests mainly in two different formats: in vitro, in vivo. That is, in vitro is basically in test tubes, okay, or your petri dish where you are taking out the cells, tissues, body part out of the whole animal and doing a test on it. Other is in vivo, that means directly into the whole animal when we are testing the drug. Okay, so that is basically in vitro and in vivo type of assay or test. Researchers determine the following about the drug. What they actually trying to determine: absorption, distribution, metabolism, and excretion information. Okay, that is the first thing. That is pharmacokinetics. That is one of the important criteria they have to determine. They want the potential benefits and the mechanism of action. Now, here comes the pharmacodynamics. So who is not aware about pharmacodynamics? I'm just going to tell you directly here. I know the other individuals in this batch from pharma medical background, you all are aware about dynamics and really you are doing good, guys. But let's refresh our knowledge. Potential, when pharmacodynamics means what the drug do to the body. That means how the drug is going to give you the benefit or the action. Okay, what is the mechanism of action? What is the dose response relationship? What is the therapeutic window? All this comes, the bioavailability of the drug. All this comes into the basically the part of the pharmacodynamics. Okay, so kinetics and the dynamics. These two are the different things. Kinetics means what the body do to the drug. Dynamics means what the drug do to the body. Okay, so these things, that is potential benefit, mechanism of action, best dosage, administration route, side effect. Okay, side effect, I'll come over here. So the first 2 points, second and third point mainly talking about the dynamics part. Then the side effects or adverse event, that also we will try to identify from here, that what are the different side effects. Effects on gender, race or ethnicity group. Okay, that means male versus female rat or mice, black versus white mice. Okay, different geographical region mice. We will try to check on them the drug. Interaction with the other treatments. If you are taking this treatment versus the other treatments also, is there any drug interaction is there or not? So we'll also see for some common drugs what are important related to that class of drugs. You will also see the interaction with them. Effectiveness compared to the similar drug. So in the same class, let's say statins are there. So we will we have atorvastatin, simvastatin. Now we have another statin in our hand, a new product for with us. Okay, we want to check that how best is our product compared to the one which are existing into the market. So we are doing a kind of a comparative study also here in the animals too. Because when we are filing the IND application, authority will definitely want to know that why you are creating this product if already some statins are there in the market. So we have to prove that answer. So these are the main things or answers which need to be ready in our hand before we are filing the IND application. Okay. Okay. Um, why preclinical study? So why preclinical study we are doing? We want to, since we are going to the human trials, we want to be ready for that. Okay. So hence we want to know the drug better before moving to the humans. We can take chance in animals, definitely. However, we should also follow ethical guidelines for animal lives also. But at least we can take chance in animals. In humans, it is really going to be a blunder that even a single death case and other things comes. Okay, it is really a big black dot on your background. Okay, when you are like having those death cases and other things. So we will definitely try our best to get more and more cases, more and more experience from the preclinical itself about a particular drug. So you can say the preparedness of the phase 1 of the clinical trials we are actually doing here. Okay, we are evaluating the product safety before going into the humans. And also we will try to, it is not overt, sorry, I'm always forgetting, allow me 1 second, let me correct it. It is over toxicity. Okay, so basically to detect the over, the toxicity is also important. Okay, because that is also like side effect at a certain level, at a toxic level is important. But over toxicity, that is also we need to check that after what particular therapeutic level or toxic level this is going to be lethal or something like that. So that is important. Then there is going to be that to establish the dose response estimation of pharmacology and the toxic effect. This is very, very, very important part which we need to know. Now, see, there is always a good example I usually give it. You have two medications. One you are taking in micrograms, another you are taking in milligrams. Okay, in B product. So if you have the particular product that you're taking micrograms, and if you are going to say that, okay, fine, like, I don't want to do the preclinical study, let me take it directly to the human trials. You don't have the dose response relationship with you, and you might start from some milligrams and you may experience some death right away because that drug is going to show the toxic effect in after certain microgram level only. Okay. But if you take the another B drug in your hand and you are going the same way without, like, with that is bypassing the preclinical trial and having no idea about dose response relationship, and you start trying into the humans, you may end up with the thing that you start from the microgram level just to be more safer side. Then after doing so many tests, you finally reach to the particular milligram level, let's say 350 milligram or 500 milligram, and the drug is going to show you the desired effect. That is also wastage of time and resources for you. So that's why to avoid all this thing in human trials, what we do, we actually try to avoid this part. Okay, in the human directly, we do the same thing testing into the animal so that we can know that what is the best dose response relationship, okay, about a particular drug, and also the toxic effect, whatever at the particular dose we can have, so that we can avoid this problem in future. Just give me 1 second. All right, sorry for the disturbance. So I'm just saying here that the dose response relationship is hence very important to know in the preclinical stage before moving to the human stage, okay, human trial stage. Another thing is to assess the drug distribution into the organ system. So where it is going more, where it is not going more. So we have already learned in our previous classes also and also in the bachelor's degree, if you are from the pharma or medical background, that there is in the brain we have BBB layer, okay, where we have filtration, that how much drug will go into the brain and all these things. Similar, in the different organisms we have a different distribution system. So that also we need to understand that how the drug is getting distributed into the body. It's a part of pharmacokinetic itself, very important part. Also, how it is getting metabolized, what is the half-life of the drug, okay, how much time it will be there in the body, elimination pathways, okay, how it's going to eliminate it via skin, okay, or via urination, via fetal way, how it's going fecal way, why, how it's going to be like excreted. So all this thing we need to understand here. Apart from that, few other special studies for other things we need to do to understand. So here, my question back to you again. What do you know about carcinogenicity? Any idea, anybody? Carcinogenicity testing, right? Anybody? A tumor? Okay, that is kind of correct, right? Okay. Yes, Sandhya, you can answer. I think you raised the hand, it is showing. You can put in the chat box. Cancer causing determined substance probability to cause cancer. Okay, okay, correct. Sumit, Shamita, you are all both are correct. That is cancer producing substances or cancer causing substances are called carcinogen. So we will check that if the product is carcinogen or not. So carcinogenicity testing. Reproductive toxicity, I hope this is a self-understood term. So that also we will test. What is teratogenic potential? Any idea? Anybody? Okay, Harshita, great. Causing mutation only in the fetus, correct. Anybody else? No? Okay, I think you all agree with Harshita. Same answer, maybe you all have. That is really correct. So teratogenic potential is basically when there is, yeah, normal fetal, its outcome finally, Nikita. Yes, so teratogenic potential means when the drug, let's say mother is taking, and that drug is actually causing the not only mutation, any changes in which the baby is going to born with some problem. Okay, so that changes, that abnormality is going to be done on fetus, okay, in the mother's womb itself when the mother is taking the medication. So that is called as teratogenic product and having the teratogenic potential. Once the baby come out, okay, the baby may have some defect, that is like a birth defect you have. So that is maybe a cause of some product which was taken and having some teratogenic potential. Okay, so that also we check over here. We will see a good example in one of the tragedy which happened in the past, okay, called as thalidomide tragedy. We will see in the history part that entire thing also. Preclinical study to be performed. So we have all this actually test which we need to perform. Okay, we do the pharmacodynamics testing, kinetics testing. We also check the effects on fertility and reproduction. Okay, local tolerance is basically like allergic type of testing, okay, of a product. Teratogenicity study, if fertile women are included, and definitely teratogenicity study we are going to do. Single dose toxicity in two species, rodent, non-rodent. Okay, so you have seen, right, many drugs, they clearly write that no information available about the pregnant women if they can take the medication or not. very limited information, no information. Reason being, this type of study like teratogenicity study or reproductive toxicity studies, these are actually not done on them. Okay, so they avoid to write that part. Single dose toxicity in two species, rodent and non-rodent. Rodent is basically our mice, rats, all those, and non-rodents are mainly mammals. So we are doing on the mammal study. Repeated dose toxicity also in maximum, like in more than 2 weeks when we are giving a drug, we are actually doing kind of a chronic testing study or repeated dose toxicity study. Carcinogenicity, teratogenicity, and genotoxicity. Here, specifically on gene, when we are checking the toxic effects. Okay, so that somehow you can consider as a mutation type of thing. Okay, so when we have those things, we do the specific toxicity studies also. Then there is acute and chronic toxicity studies. Now, what is acute toxicity and what is chronic toxicity? Anybody? Some individuals in this group are really active, believe me. Others are maybe they are like not answering much. So acute is short term. Adeline, very correct. Anybody else? What is chronic study? Long term. So others, do you agree with that thing or you have the vice versa answer? So I just take the silence of the others as okay. So Naila, chronic is severe. Okay, agreed. Chronic is like life threatening. Repeated exposure, chronic, agreed. Correct. So great. So all of you are some way or other way correct. But again, few things. Chronic is not life threatening. Please keep this thing very much clear. Okay, guys. So whoever has the same answer like Sneha. So chronic is not life threatening. And also one more thing was there. Chronic is severe. So severe, no, chronic doesn't mean severe. Okay, severity is a different aspect. Severity means intensity of any specific disease. We are not talking about intensity. Okay, and life threatening also somewhere you're talking about severity part only or seriousness actually. So those things are not related to chronic. Chronic as as Adeline just mentioned in the beginning, and the others also agreed with that part. This is basically acute means short-term studies like 1 dose or 2 dose you would give and you want to check quick things, okay, like toxicity, okay, like a particular mechanism of action, okay, distribution, absorption, all those things. So here you can do the acute studies. Chronic studies you do for long term when you are giving your medication more than 2-3 weeks, okay, and continuing for 1 year, 2 year giving this medication to see that if the drug is giving any long term side effect, any carcinogenic effect, okay, any this teratogenic potential it have, all this thing or any other such long time type of side effects are there or not. So for that we do the chronic study. There are basically, again, the studies can be divided into acute or chronic study. Okay. Okay, now I think we can move to the clinical trials. Any doubt till now, till the preclinical trial part? No? Great. So brilliant students. Okay, great. Okay, clinical trial. So let's start with that definition part itself. Okay. So if I just go with the general definition, okay, general definition is a layman term. If you ask me, I can say it is basically a well-designed, organized research where we have everything on paper first, okay, on the human beings, okay, which is intended, why we are doing this thing, to intended for getting more information, adequate information about a particular therapeutic agent. for its safety and efficacy part. When we are doing this thing, we are doing the clinical research. Again, let's summarize the entire thing. We are testing on the human bodies. Okay, human trials are going on. It's a well-designed, organized research. We are not doing in just like a manipulative way or something like let's give and test around. It is not like that. And everything is as per protocol. Protocol is getting approved first with the authority, and then only you do. Okay, you go step by step. Why we are doing all this thing? To get the adequate information, more information about the particular therapeutic agent, the drug, especially on its safety and efficacy. So this is basically a layman term definition to explain anytime if it is asked for clinical research to you. And as per the ICH GCP guideline, that is another international guideline. Now, what is ICH GCP? ICH is basically International Council on Harmonization. It is an international council with several global countries. They are actually part of it, and they try to uh, create some harmonized guidelines in the clinical research, which can be followed by all over the world, whoever are the members, so they can follow. Of course, country have their own guidelines, but they can follow and take some reference from these guidelines. So these are internationally approved guidelines, out of which ICH, under ICH there is a GCP, good clinical practice guideline is there. As per that guideline, there is a definition. So this is a technical definition, you can say, which is available of the clinical research. So clinical research as per this guideline is any investigation in the human subjects intended to discover or verify the clinical, pharmacological, or other pharmacodynamic effects of investigational product. You need to have these three things. Plus, to identify any adverse reaction, that means side effects, to any investigational product. Plus, to know more about or to study the absorption, distribution, metabolism, and excretion, that means pharmacokinetics of an investigational product. Okay, and all this study why we are doing with the object of ascertaining its safety or efficacy. That is the same thing which we have just discussed in the layman term, but here it is completely technical, covering each and every aspect over here, so that there should not be any miss to consider something as a clinical research or something not as a clinical research. So when we follow this entire definition, you can say that we are doing a clinical research on a drug. Okay, one more thing, clinical research or the clinical trial, clinical study, human trials, all these are synonymous terms. So if you hear any of this term, please understand that it is talking about clinical research only or clinical trial only. Okay, these are all synonymous terms. Any doubt on the definition part, anybody? Okay, now moving to the objective part. So objective of clinical trial. So what is the objective of this clinical trial? Basically, two main objectives. First is to get the scientific evidence on the safety and efficacy of a new therapeutic intervention. So we want the scientific evidence, which we are going to present it to the authority for the approval. Okay, so that is important with one objective. Second is to follow the well-designed clinical study to generate the meaningful result. Result can be positive or negative, but we follow the well-designed clinical study. That is another objective. We are not just doing in a what we used to do in our 12th or 11th classes, or in our bachelor's, right? They're doing some practicals, doing in a manipulative way and taking out some result and submitting it to our teachers to get the full marks. This is not like that. Here we have a clear-cut protocol, approved protocols, approved other documents, inclusion, exclusion criteria. As per all those things, we finally do the clinical study. Okay, so and the result should be some meaningful result should be there, not any haywire results. So this is mainly, these are the main two objectives. Now comes the clinical trials done for. Clinical trials done for the new drugs. Any new drugs you would like to create, you need to do the clinical trials. New medical devices. Okay, the drugs can be your biologicals, your vaccines. Okay, the drug itself. Okay, that's all it actually includes in the new drugs. New drug delivery systems. Okay, and the new surgical procedures. Our focus is more on the drugs right now when we are talking and specifying, but for all the others we also need to do the clinical research. Here I would like to take your attention to two things. One is new drug delivery system. So for that also we have a particular product. Let's say we have diclofenac in the market as a ointment or cream or lotion like that, right, for pain relief. However, you have seen that diclofenac also comes in the tablet, as a capsules, as transdermal patch, maybe not in India as a transdermal patch, but in other countries. So the same product can come in different delivery system or formulations. Okay, so we are taking orally versus transdermal versus it can be rectal versus it can be IV. There happens that there comes a few one drug in the different drug delivery routes, also in different formulations. For all of them also we need to do the separate research, and then only when we prove we have to get the particular authorization. So maybe you have one authorization for a tablet, maybe next time you go for a particular transdermal patch of the same drug, you need to run the clinical trial again and prove it for another drug delivery system and get the new product approved. So that's a cycle which goes on if you want new drug delivery system. Also for the new uses of the existing drugs. Example I have given you minoxidil. Same example we can take. Okay, that you got the minoxidil early in the market for blood pressure related disease. During that market, you come to know that it is also showing some problem side effect of hair growth. Now you captured that as an advantage and or as an opportunity, and you did some clinical trial research again, and with a good number of evidence, scientific evidence, you put up your file to the authority that you want this product in the form of another brand name for hair growth product. So that can also happen, okay, with the same particular product. So we have to do the clinical trial for them also. So for all this we need to do the clinical trial. Any doubt on in this part? So in the nutshell, if I would like to say, that basically this entire clinical trial, this what we actually do over here, we are collecting the scientific evidence for safety and efficacy of a new therapeutic intervention, or you can say the new drug delivery system or the new use of the existing therapeutic intervention. So that is the clinical trial all about in nutshell. Now, how mode of delivery of a drug determine? So how we do this thing is actually it is another drug discovery. It goes to the discovery when we are focusing on a particular target. Pratiksha, for your question, when we are doing the discovery part in the very beginning, when we are finding the target, okay, at that time we will see that what is the best mode of drug delivery system. Okay, scientists will decide on the basis of how best they are getting the result from various different delivery systems. Okay, but when your product is in market, okay, at that time company, to be frank, very much frank to you, as per industry, okay, industry are also not fool, they are very smart. In the beginning only they know that the drug can be given from various routes, and one route is better than another route, okay, and then it is better than the third route also for administering. What they do sometimes, they take out the medication with the from the third route, which is not the best route, and after some time they just put up the another, like route of administration application that they want also to try. Let's say first time they put it as an transdermal, then they will just say that on another route of administration we would like to give this medication for the very fast action is the oral. Then they will also prove with evidence that they want to give it to the IV. So that is a smartness company also plays, believe me. Okay, not all the companies, it also the authority also checks a lot of time, but company usually plays this thing. How they decide this thing? They decide in the very early stage during the drug delivery or discovery only, that which route of administration they want to go with. Plus, with the help of preclinical studies, they actually prove that themselves also that this is the best drug delivery system. Okay, the test and other things, Pratiksha, that I cannot provide you right now because this goes to the research part right now. This doesn't come under the clinical trial. In the clinical trial, we already have that this is the route of administration, this is a drug. We need to test into the human trial. Okay, so I hope I answered your question. All right, clinical trials in a nutshell. So I'm just taking you right now as a very high level. Okay, right now, it's before we go in depth, let's understand a little bit from a very, very, very high level, like how the eagle looks around the entire area from the very top. I'm just taking you at that level to see that what is going on in the department and how the entire trial runs. Okay, so first is approved protocol. Protocol, what is protocol? It is basically a document which is having everything written over there, okay, in the document, which you need to do, what you need to do, everything is there in the protocol. From the protocol, we can create our other documents also. So you can just like a master book, which we create early. Sponsor usually create this thing, okay, with the help of other coordinators, doctors, okay, but the responsibility is for the sponsors. Once that is done, then there comes the investigators, the doctors, who is going to take care of the trial from here on. So they also check the protocol and other documents first before sending it to the authority. Okay, then if everything is fine, they will send it to the authority that this is going to be the protocol, like doctor and other things, okay, everything. Once that is done, authority convinced with that part and give you the approval, then you can start recruiting your patients as per the inclusion exclusion criteria. That means who is going to be included and excluded. Okay, as per that, you are going to do the patient recruitment. After the patient recruitment, you are going to start doing the study. Okay, so when you start doing the study, okay, on the humans day-to-day basis, you are going to generate a lot of data, right? At day in, day out, so much of data you will generate. All the data will start going into the computer system. Our CDM team actually comes in picture. Okay, so that data management team starts working and feeding the data, compiling the data into the computers. Then comes the biostatisticians also, who started running in parallel to finding the statistical graphing, charting, all these things, so that the result can be provided much better. So with the help of all this thing, we are actually ready to present some publications, reports that what is going on in the test. And hence you have seen in so many journals that so many clinical trials or tests which are ongoing, the company started publishing their reports in journal, so that you can be aware that which drug is on the way right now. And hence, because of this, we got to know about so many updates on the particular upcoming drug, like COVID-19, this drug is in this phase or this phase, this is showing positive results. So here, by this way only we come to. Once this everything is done, we file the new drug application or marketing authorization application. Whatever you want to say, it is the same thing, that is authorization application we file to the authority. With the help of this application, the authority reviews your products and give you the approval if everything goes fine. Okay, and then you can celebrate your success of this big project. So that is the entire journey of the clinical trial. Starts with protocol and ends with the approval of your application. Okay, so this is from the very high level. Now we slowly go in depth in each of these stages to understand them. Okay, CDM and statistics, actually it is completely a different domain or a part. Of course they are supporting in the clinical trial, but it is completely a separate part. Okay, it's a separate domain to understand and to have an expertise in these things. Okay, it is not a laboratory task. Okay, right now what we are understanding in general is basically, definitely we'll try to see these tasks also, but in general we'll try to understand the first few steps, like what we are actually, how we are creating the protocol, and what is like an investigator, their responsibilities, other stakeholders, we need to understand them. Okay. Any questions, let me know in the chat box, please. Clinical trial is a summation of good clinical science, good statistical design, sound ethical conduct, and integrity and quality. So when we have all this together, okay, we can say that we are doing really a good qualitative clinical research. Okay, when we have good clinical science knowledge, we are following everything ethically, and statistical design also, everything is going very correctly. We have the integrity and the quality in it, then it is actually showing that our clinical trial is going correctly. So allow me just a minute. I'm looking that if I can take a break now. Allow me 1 minute. Let me take you a little bit ahead. I have to just check the slides. Okay. So I'll just complete these 2 slides and then we all can take a break. Okay. Why some participate? Why some do not participate? That is important. Okay. Why some participate? It is because there are many reasons. One is they want to give back to the society. Okay, the participants, they want to give back to the society because they are a part of this public. Okay. Secondly, another reason is maybe, like, let's say the cancer patients are there, or there are other patients who tried the other drugs and like whatever the available medications in the market, but they all, they are not satisfied with them, exhausted with all other options. They want to try this new treatment, new healthcare service. Okay, so they also sometimes just in the mind that similar like the second option, they just want to try some new service, new drug. Of course, the few individuals, they just run for the payments and incentives. Maybe not much in the Western countries, but of course in India, in African countries, you will find a lot of individuals who actually participate mainly for payments and incentives. Okay, for them, for these aspects, they want to participate. Second, some individuals, they think that there is a better support, better supervision in the clinical trial. Hence, they participate in the clinical trial. And then there can be other reasons too, maybe not listed, but definitely maybe you also have. So if you have any other reasons in your mind right now, please write in the chat box. Okay, I'll take up that reason to like why also you are thinking that there are some other reasons for participation. Okay. Another thing is why some do not. Definitely there are so many like people, if you ask them, they will say, no, I don't want to participate. Okay, why? One is mistrust of studies. They are like, not trusting much on studies. Just go back and think in the beginning of when there was Covishield vaccine medications and other medication studies were going on. Believe me, if I start asking you also, you will say no, 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 I don't want to participate. Who knows that how this medication is that going to do the clotting or something else or something else or something like that. So, and they don't want to trust the studies also, that like, if it is going to be done in correct way or not. So there can be mistrust of studies. And yeah, you do not want to become a guinea pig, right? You want to be a trial tested medication, but you don't want to be a part of trial. You don't want to be a guinea pig. Okay, that if something happens, it happens on you first. That is another reason. Do not meet the criteria. There are always inclusion exclusion criteria in the clinical trial. Inclusion means, let's say that, like 30 years to 40 years of adults, males, they can participate. Now if a female wants to participate, it is not allowed because there is not an inclusion criteria, so you cannot be a part of it. Or a person or a male of below 20 years of below 30 years of age, he or she they cannot participate. Okay, that's the inclusion criteria. Same way the exclusion criteria is there. So it is kind of an opposite also of the inclusion. Okay, plus there are some other criteria. So let's say 30 to 40 is okay, but if in 30 to 40 years of age, if the patient is suffering with some cancer problem or let's say the blood pressure problem, he or she cannot participate, even though he or she is 30 years old. So that is exclusion criteria. So if you are not fitting with that criteria, you cannot participate. So that is another reason. Okay, cannot give up time for study visit. That is another very important reason for now in this busy world. Okay, we don't have much time to go back to the trial site and take the drug in the supervision and all this thing. So that is another problem. Long distance is another barrier. And we have if you are living in a particular one region and from there you have to go to the 10 kilometer 100 kilometer and you want to for the test purpose, you don't want to travel this much. So that can be another barrier. And we cannot keep the clinical trial in a van and take to your home and do the testing. It is not also possible. So that is another barrier. So these are some of the reasons because of which some want to participate and some don't want to participate. Anybody else has any other reason to participate and not participate? Anything else you would like to add on? Yes. No? All right, so great. So try to remember this thing. The question can be there in the interview. This is another important part. Participation in the clinical trials. Just this slide, and then we will take a break. So participation in the clinical trials, basically diseased individuals as well as healthy volunteers can participate. Definitely, it depends on the particular phase to phase of the clinical trial and as per the inclusion exclusion criteria of the trial. So they both of them, diseased individuals as well as the healthy individuals can participate. Depending on what questions in the trials are designed to address and what kind of trial you are considering, the participation is there. So if it is a particular specific to any cancer stage, let's say stage 2 is there, so that is one of the criteria to participate, and that is a kind of trial is basically a cancer related trial. So definitely the one who is not with the cancer, they will not able to participate. And there are some questions in the trials which we need to answer. Okay, questions means not the participants need to answer, but as a clinical trial team, they need to answer that if this drug is going to work on cancer stage 2 or not, with a particular with a particular some other comorbidities if the patient have, if the drug is going to work on that also or not. There are some related questions are there on the basis of which we create our inclusion exclusion criteria. So you can see the inclusion criteria. So it's like eligibility or inclusion criteria. So these are basically eligibility criteria. These are the characteristic of volunteer required for participation in clinical trial. So like age. sex, stage of disease, previously used treatments. These are some of the factors, okay, which we keep in mind, where we are putting some barriers, some age barrier we are putting, sex barrier we can put, male, female, okay, like that. Stage of disease we can put. These are the barriers we can keep in the inclusion criteria, and also similar barrier in the exclusion criteria. Exclusion criteria are the characteristic that mean that someone should not participate in a particular clinical trial because of a particular exclusion criteria. If you are fitting with the exclusion criteria, then you cannot participate. Researchers use this criteria to identify the most appropriate participants. Okay, that is the main reason why we need the inclusion exclusion criteria, because we don't want to use the human as a guinea pig. Authority don't want you to use that way. We have to specify the particular inclusion exclusion criteria and get only those subjects, those participants, because from them only you can get the best data about the potential benefits and the risk of a particular product. One can check the eligibility criteria in clinical trial registries. That is important. So by the end of this session, or somewhere after 2-3 sessions, when you are you know the things better, okay, after the designing part, clinical trial designing part, I will take you to one of the important international website, okay, in which you can anytime check so many clinical trials which are ongoing, and you can easily understand the trials. Because once you have a good knowledge, then you can start understanding that part. Okay, so I'll take a pause here. Let me see, it's almost 11:31 in my mobile, if I'm correct. Same thing with you. So let's take a break of 5 minutes or 7 minutes or so. Okay, and then I will continue. Or allow me just 1 minute. Let me cross check 1 second. Ne, ne, ne. Okay, guys, so we'll just take a break of 5 to 7 minutes and then we will continue. Okay, so you can take a bio break, quick bio break. I'll just stopping over here. Hi all, let's continue. Sorry, it was a little bit a big break. Now I will take you through a brief historical review. Again, this is not a main history part. I will take you through the historical part. But I think by the end of this session, after this presentation, or if it is the time allows, otherwise tomorrow we will complete that part. It's in brief. If I would like to tell you that how this clinical research started and slowly progressed with so many guidelines and other things. And that like 19th century somewhere, so many ancient pharmacy practice started. Okay, not much clinical trial at that time. And when they started doing this type of trial, it was actually on the hit and trial and error basis type of things. Okay, so what nowadays we are doing in sometimes in our laboratories, right? We are just doing some trial and error and producing some product in our 11th 12th classes and in our bachelor's also to get some kind of result. So in actual practice in the clinical trial, that used to happen in early 19th century somewhere, and the practicing physician decided it's actually after that I would just like to complete this part. So practicing physician, they decided to go through all this type of trial and error basis type of test, okay, with not much guideline in hands. Okay, in the 20th century when we came, there was a huge expansion of pharma industry actually happened. With that, so many guidelines started coming up slowly. Again, not very fast in the early 20th century, slowly. Definitely, there were few incidents happened in the human history, which gave slowly some important guidelines. One of them is 1947. There came the Nuremberg Code. Now, what is that? I will let you know tomorrow or in the history session actually in the detail that what is Nuremberg Code and what is the what Nazi physicians they did in the Germany. After that in 1962 there comes the thalidomide disaster. Okay, that also gives so many guidelines, so many things. Very important milestone in the clinical history. Then Declaration of Helsinki, which happened, which came actually because of this thalidomide disaster only. So in the Helsinki, it's a particular geographical part in the world. There they have finalized some important guidelines called as Declaration of Helsinki. This World Medical Association, they provided in 1964. Okay, they provide some important guidelines which need to be followed for the ethical conduct of clinical trials. Then comes the slowly when the year passed on, okay, most of the countries have started putting emphasis on the informed consent. That means where the patient can provide his or her own consent that he or she is okay to go in the participation. There should be no forced participation. So that's how slowly our entire clinical trial history progressed. And by the time we reach to the 1990s or so, we have so many guidelines like ICH guidelines and FDA guidelines and other country-specific guidelines, which are more rigid and a lot of such steps to take care of the patient's patient ethics or ethical conduct of clinical trials. So there are few guidelines. Some of us we have already either read earlier, like Schedule Y, Drug and Cosmetic Acts, 1947 in India, Indian GCP guideline, another important thing, which I actually expect you to read because in this short session, okay, few hours of session, we cannot complete the entire guideline. It is entirely a separate thing. It takes time. But definitely after understanding this entire course, it would be quite easy for you to complete that guideline. So Indian guideline for the clinical practice, apart from that, our ICMR guideline in India, internationally WHO guidelines available for clinical trials, mainly ICH GCP guideline. So that is another very important guideline at the international level. Okay. So these are the various guidelines which are available. Okay, some of these we will definitely try to cover up or see in our this course, but not able to complete all this because this will take completely entire vast amount of time. Okay. Stakeholder in clinical research. So again, the clinical research is not an in silo project. Okay, you cannot just sit in one department and say that this project is going fine. Okay, like various other departments of pharma industry or life science industry, clinical research also have various stakeholders in and around it. Okay, it is like sponsors, investigators, the study participants, monitors, auditors. IRB, IEC, I will tell you in the another session where I am going to tell you in detail about IRB and IEC. This is actually a body, okay, which is a body, important body in the clinical trial. I'll let you know in the next session. That is stakeholders' roles and responsibilities. It is one of the important stakeholders. Then biostatisticians, our CDM people, data management people, okay, regulatory affairs and the regulatory authority itself, reporting and documentation, medical writing teams, business development groups, project management teams. Then I think I covered most of them. Our clinical site, CROs. Okay, these are all very important stakeholders. We need all of them somewhere or other way, okay, to complete the entire project of the clinical trial. They all either influence in a positive or negative manner to our entire project. So we should take care of these all stakeholders in the clinical research. Now comes the types of clinical research. So there are various types of clinical research available. Mostly when we are talking, okay, in our mind we keep the first one, that is treatment research, in our mind. However, there are various other types of research, clinical research available. So it is again as per the FDA, this classification is available. Maybe you will find the different classifications if you start searching the Google and other. But let's understand some international classification as per FDA. So there is treatment research, which we know in general, that is when we are giving an intervention to the patient, okay, or a medication, physiotherapy, psychotherapy, new devices, vaccines, all those things when we give, okay, to basically, or even a surgical procedures to the patients to understand how this particular product is going to work, okay, and treat the patient. So this is called as treatment type of clinical research, which we actually know as a clinical research, that is a treatment research. There are other research are available like prevention research. So here we look for better ways to prevent the disorders, prevention, okay, not treatment. So here we are usually looking for the research on many medications we use for preventing something, not treatment, okay. Vitamins, yes, vaccines, sorry, comes over here because it's a preventive measure. So vaccines research comes under the prevention type of research. Vaccines, vitamins, minerals, lifestyle changes, a lot of different research related to the prevention of certain disease. That is called as prevention research. Another one is the diagnostic research. Now, what is diagnostic research? It refers to the practice for looking for better ways to identify a particular disorder or condition. So this research is mainly, it's not like treatment or the prevention. It is for diagnosis purpose. So when we are taking out some drugs or some compounds for diagnosis purpose, okay, to find out the particular disorder in the early stage. When you are doing that particular study for that particular molecule compound, that is called as diagnostic research. Screening research aims to find the best ways to detect certain disorders or health conditions. Okay. So here we are actually identifying the methods basically by which we can identify the disorder or the condition in the early phase. So we are trying to identify those methods, those ways to detect the particular disorder or health condition in early phase. Here we are not actually using the specific drugs or molecules or compounds. We are actually identifying the best methods. Now, If you ask me that over here also we will go with this and this and this phase inclusion exclusion criteria, I can say yes, but not in the same way what you have in your mind related to treatment or prevention type of research. Okay, this is more or so it's like an observational type of studies we usually do in such method. Quality of life research explores ways to improve the comfort and the quality of life of individuals with a chronic illness. So I hope you all have seen that in the newspapers, in somewhere the last or second last page before the sport page where the science section comes. Okay, there sometimes comes the articles where it says that you have to consume this and this vitamins every day or this and this, or you do not have to like do a smoking not more than this much, or alcohol or anything else, okay, which is showing that how your life is going to get affected in long run, or your quality of life is going to be affected. So from where this data comes, this is from the quality of life research. Again, this is also kind of observational type of studies where they are observing for long time, okay, with particular method they will try to generate the data and present in front of you. Then comes the genetic studies where they aim to improve the prediction of disorders by identifying and understanding how genes and illness may be related. So you can say it is more also clinical trial on genes. Okay, they will try to like explore that part. Research in the area may explore ways in which a person's genes make him or her more or less likely to develop a disorder. So you have also seen this thing, right? That a mother, if she has a particular this type of gene, or a father has this type of gene, there is a good possibility that the baby is going to have this disorder or may not have this disorder. So this type of study, which is actually going to prove this thing, these are called as genetic studies. Okay, this may lead to development of tailor-made treatments based on the patient's genetic makeup, on the basis of which, you can tailor the entire treatment if we know this thing early. Epidemiological studies, it is basically to identify the patterns, causes, and controls of disorders in the group of people. It is like a case study when we are doing, okay, for certain group of people. In this particular region, this many peoples are actually residing, and they have a pattern of drinking this water or that water, or taking this medication, or so many things which they want to identify a pattern in a particular group of people, on the basis of which they take out a particular decision. These are epidemiological studies. Okay, here we are trying to identify a pattern, the cause, and hence we try to identify a particular disorder. Okay, these last few studies which you have seen are more or so are basically observational type of studies. Prevention and diagnostic is completely different. Diagnostic is basically where you are not actually identifying in a specific a compound or so. Sorry, diagnostic is something in which you are trying to identify a particular compound or so that you can identify the disorder. Okay, refers to a practice for looking the better ways to identify a particular disorder. That you test, right? You go in the laboratory, you identify certain molecules that when you put in this like this in the test tube, and you will find the disorder is there or not. So you try to find those agents or those molecules which can identify the disorders. Those research is related to the diagnostic research. Prevention is separate. Prevention is for your disease, okay, to prevent the disease. That is vaccines. Okay, take vaccines, very good example. When you are identifying this disease, this Covishield, Covaxin, what this company clinical trials, we can name them. These are preventive type of clinical research, not treatment. Other companies, what this DRDO took out this medication, or DRDO, or the other medications which are coming up in the market, okay, with the companies are in somewhere in the clinical research, those are treatment. Now, similar way, the diagnostic is different. Diagnostic means how you can identify the disorder. Identifying the disorder, you need certain agents, okay, certain methods to identify. So those are called as diagnostic research. Okay, so who is that? Shamita, I think you got the idea now. Okay, I'm sorry, Pratiksha, if I'm correct. Yeah. How epidemiological study different from clinical? See, clinical study is a big, name. Under this, these are all the clinical study only. Please don't get confused. These are all the types of clinical research or clinical study. But there are different types of clinical studies. You can do the clinical study in the lab or outside the lab too, as an analytical, as a randomized process, okay, in the lab or outside as observational type of study. The last few, apart from genetic, are mostly observational type of studies, where we are observing a certain quality of life. Okay, by the help of this, we are actually, or epidemiological study, a certain pattern in a particular group of people, how Indians have the different behavior and other things, okay, towards this particular drug and all this thing, okay, compared to the Americans or compared to the Chinese. Hence, you are actually doing an epidemiological study and you provide a particular result. So that is also a part of clinical study only. You are doing a study in humans only. But this is an epidemiological study. You are not doing a treatment. You are not giving a treatment to a particular patient. It is not an interventional type of study. It is a non-interventional study. Okay. Okay, great. Now moving to the clinical phase. Now moving to the clinical phase trials, phases of the trials. That is phase 1, phase 2, phase 3, like that. So let's a little bit understanding of those phases also. So what is phase 1? In the phase 1, basically we target 20 to 100 or maybe up to 300 or 1000, depends on your budget of the project. Okay, we target this many people and mainly healthy volunteers for a short span of time, not much time, 1 month to 3 months or maximum 6 months it will go. In that we mainly try for safety studies. We want to know the safety of our drug. Definitely efficacy is there in parallel, but mainly our focus is on pharmacology part, clinical pharmacology and safety of the medication. Okay, or PK, PD, all the studies we would like to know, because we are giving in healthy volunteers, so we can have a good response and everything. So we are targeting on that part. Phase 2 is basically 100 to 300 patients, or it can go up to 3000 too. Okay, it is mainly in the diseased individuals. Okay, so the diseased individuals, in that why we are doing, because now from the phase 1 we know about the dose response relationship, safety and everything. Now let's understand more about efficacy. And from where we got the more efficacy data? From the diseased individuals. Okay, so here we are targeting them. The inclusion criteria is going to be more towards the diseased individuals. If it is cancer, let's say cancer stage 3 people we need for this one. Then after that, when we have a good data of safety and efficacy both from these two stages, it is time to go for the multi-centric level. At the various different centers and in the bulk of population, we want to try our drug. Okay, to find the rare of rarest side effects and the safety parameter also a good data on efficacy. So here we can range from 300 to 3000 to 10,000 also, depends on your project budget. Okay, and in various centers, it can be, let's take India as an example. It can be one center in Ahmedabad, one in Delhi, one in let's say in Shimla, one in Kolkata, one in Patna, one in Hyderabad, one in Chennai, one in Bangalore, okay, one in Mumbai. It can be anywhere, okay, as per your company budget and as per the link. It can be so many centers you can provide so that you can target different populations, okay, to understand better about the product. So hence we have the more data from this stage. Again, the main thing is safety and efficacy. We want more and more data in this stage. Once this is done, we will file the application regulatory submission. Phase 4 of the clinical trial is there, but phase 4 itself is not actual clinical trial. Phase 4 means post marketing, whatever it's going on, we will consider it as phase 4 only. That is also called as post marketing surveillance, means keeping an eye on the drug. Okay. There are various part of the phase 4 like pharmacovigilance is there, pharmacoepidemiology studies are there. Okay. And there are many other things which we do in the phase 4, you can say, or post marketing stage. But we will try to limit our knowledge till this phase mainly. This is something which I have just mentioned. One more thing, the phase 2 you may find in some books or maybe in some studies when you start reading can be further bifurcated into phase 2A and 2B. Now phase 2A, what is that? Phase 2 is basically in in phase 2, the 2A part is actually to understand the best dose or dose relationship. Okay, that is 2A. So if we still want to bifurcate two phase into two parts, we can say 2A is mainly to identify the dosing requirement or the best dose for a particular diseased individual. 2B is when we are giving that particular best dose to see the efficacy. That is 2B. Okay. Phase 3 is 3A and 3B. It can be there. And of course, in most of the papers, you will find 3 and 3B. 2A, 2B, we will not find much, but 3A, 3B, you will find. Why? 3A is basically what you know about phase 3 itself. It is actually 3A. That means whatever the multicentric trial studies you are doing, okay, in large group of people, that is what you are actually doing a 3A study. Okay. In 3 and after 3A, by the end of 3A, you are ready to file your application, NDA application, okay, to the authority. After that, during this time when your application is in process, that is for 6 months to 1 year, 1.5 years, or 2 years, okay, what you are going to do, you cannot sit ideal, right? You are started preparing yourself for the launch of the product. And launch you cannot do just saying that I did this research and this is my result, please take my drug. We need to showcase as an advertisement that how best is my drug compared to the other competitors, right? And some other extra information about other drug-drug interactions, other food-drug interactions. So that extra time we use, okay, is called as extension studies. So we are basically doing more study on our drug, especially the comparative type of study with other marketing competitors, other food drug interaction, drug interaction, all this extra information we want so that we can put this information during our launch. Okay, so that is called as extension studies or 3B. Of course, if you find any such data which is which is side effects and other things, you keep on sending it to the authority keep on updating your file. Okay, so that is okay, it's perfectly fine, but we need to utilize this time as a phase 3B. Any question on these 4 phases? Okay, so let me okay, so after this I'll take you to another matrix part, that is this big matrix you can see, phase 1, phase 2, phase 3, phase 4 on the column side, and on the rows you can see objectives, and these there are different factors are written over here. So again, I don't want you to memorize everything, but I request you to go through with this all the time. Since you are all interested in clinical trial, these are important factors you need to remember. Okay, so objectives, as I've just mentioned, the same objective you'll find here or there, a little bit change and twist in terms. Okay, and it is more or so of as per FDA guidelines, this entire thing is provided, but definitely it is available in the same way for other countries. We also follow the same thing. Okay, you need to check the objectives for each phase, that why it is there, this phase. factors to be identified. What are the important factors that we need to understand? Okay, like in the phase 1 we need to understand the bioavailability, bioequivalence of the product, dose response relationship called as dose proportionality, and then metabolism, pharmacodynamic, pharmacokinetics. These are the main things which is important factors we need to identify. Same thing in phase 1, data focus point is there, like vital signs we are looking, those safety related issues, if there is any side effect comes, a vital sign showing up, plasma serum level of drug, and adverse events. Okay, so we'll see all those signs and symptoms and safety issues. Design features. So designing, if you like, and if you don't know right now, please don't go with this thing at this moment. Design features, once we complete our designing lesson chapter, then you can come back and easily understand this part, designing part. So don't go in much depth here, blinding, unblinding, and all those things. I will explain you in the third or fourth presentation somewhere, and then you can go back and check this out. It would be easy then. Duration, you all can understand that how much time it will take. And then population, that also we can understand that what kind of population is there in as inclusion criteria. Sample size, what is the sample size at a minimum? This is minimum sample size, maximum can go more than that. Okay, and some example are written. If you understand, that is good. If you don't understand, don't go much in depth in the examples at the moment. Okay, so this is how you have to check it out. So when like you start reading this entire thing, please make sure that you start like understand this entire thing over here, and if there's any question, let me know. Now, a few blockbuster drugs which get withdrawn from the market. Okay, so these are, now why we are understanding this thing, because after even so much of our like inputs, efforts in the clinical trial, still there is a possibility that your drug can be withdrawn quite soon from the market. Yes, of course, if it happens, we are actually in the lose-lose situation. We lost a lot of money, lot of effort, lot of things already. So that is the another important thing which we need to know. So there are like, and how, why this happened? Because definitely sometimes it happens that we tried our best, but there are some side effects which we either ignore during the clinical trial to consider that as a big problem, and that actually makes a big problem once it comes into the post-marketing stage when millions of people start taking the drug. Okay, so some examples are here. You can take some examples from some of these examples and remember, memorize them also. Okay, thalidomide 1961, when it like can actually get withdrawn in this year, and reason being the teratogenic potential, that is congenital limb defects happened. Okay, baby born with the limbless or some limb defects. Binoxaprofene in 1982, another milestone, important. Hepatotoxicity actually created. Okay, it's a US product, and it created hepatotoxicity, because of which it gets withdrawn within next 3 years. Finformin 1982, lactic acidosis. It get withdrawn from the market. Similar like that, others are there. You can see fenfluramine, and you can just come a little bit in the bottom part, rofecoxib, valdecoxib. These are coxips, right? It's our NSAIDs. If you are from the pharma medical background, you all know these are common pain medications. Still, these also get revoked or withdrawn from the market because of cardiovascular events or like nephrotoxicity, all these reasons. So all in all, here I would like to say that it's not possible, it's not always there that you put your effort, you put your money in the clinical trial, everything, so the drug will sell in a good way into the market for next 20-30 years. There can be a possibility because of our lack of a check during the clinical trials, okay, on a particular side effect or ignorance of that thing, okay, that some side effect can be actually get amplified during the post-marketing stage, or if some side effects which we have never identified that can appear,
okay, as a new side effect in the post-marketing stage, and can be so severe that your drug need to be withdrawn. Okay, of course, the pharmacovigilance department that actually starts working mostly in the post-marketing stage, they try their best to take care of these situations, but there can be conditions of, like, you know, like these many drugs, they can be revoked. There are some examples which I would like to take you through. Statin is there. Okay, Statin is one of the good molecule, okay, from Bayer company for the cholesterol. Statin, by the name you can see, it's for cholesterol mainly and prevent cardiovascular disease. So company take out this product to compete the atorvastatin Lipitor product, which was from the Pfizer and selling like anything. Believe me, Pfizer earned a lot of money from atorvastatin. So in comparison, the company tried to launch this product in 1990s, okay, late 1990s basically. But in a few years, okay, this product in a couple of years actually, by 2001, the product get withdrawn from the market because of the fatal rhabdomyolysis. We know that rhabdomyolysis is one of the important side effects of statins. Okay, but over here with this drug, there are cases of deaths. Hence, the finally authority asked the company to withdraw this product. So they lost millions of billions of dollars and everything. Okay. Another good example is rofecoxib. Rofecoxib is another very important product. Okay. Merck company, one of the best product which Merck ever took out in the market. Okay, with a very good hope that it will sell like anything in the market. Like it actually did in first 4 or 5 years. It really earned a lot of business from the market, from US market, and it is actually sold for the osteoarthritis, acute pain conditions, and dysmenorrhea. Okay, widely used product at that time. Okay, almost you can say the sales revenue of the product was $2.5 billion. With the brand name of Vioxx, it was sold 2.5 billion in 90s. Okay, you can see in 90s, not today. It also get approved with other brand names like Siox, Ox, like that in different countries. Okay, however, it get withdrawn by 2004 September 30 after getting so many reports of the heart attacks, strokes after using for long term. Okay, which maybe not clearly studied during the clinical trial, or maybe ignorance, something happened that actually because of this problem the drug gets withdrawn. Now here I would like to tell you something which is offline information about this product. So as per my understanding, what I have heard in this industry, this company Merck actually came to know about this problem within like a 1 year or so. Believe me, this happened because of our pharmacogenetics department. They started knowing this thing. They tried their best to avoid this problem or minimize this risk. But since this is related to the heart attacks and strokes, that means death, which is available with that, it is really, really hard for the company to hide out this thing. And at least they tried their best to sell the product for a few years. So finally, because of whatever their linkage with FDA and government on all this thing, this is offline information available. They tried their best to sell this product at least for 4 or 5 years, still after getting awareness about the first 1 or 2 deaths in the very early year, 1 year only. Finally, after so many deaths and other things, company, it is asked by the authority to revoke the product. So this happens, okay, this happens. So it is not possible that your clinical trial is going to be always successful and your product is going to sell for next 20, 30 years. It happens that it can be revoked in the early stage. Okay. So, but definitely as a clinical trial personnel, our like, effort would be there always to find out more and more side effects and other things so that we can announce that thing early in the stage only, that these are the side effects available. Okay, there is a summary. So clinical research is an integral part of drug development, and designing, conducting, monitoring, appropriate quality assurance, all these parts are their important parts. We will understand in upcoming presentations about this. So from the beginning part, where we design, where we conduct all the clinical trials, where we monitor the trials, quality assurance, everything is so much linked up, and everything, even the data management, statistics part, everything is so much linked up that if any chain is going to be loosened, then we are going to be in big problem. So all these different parts are so much important for the success of the clinical research project. Okay. Now, few other important terms and things are there to understand. I will also share with you, like, if I'm correct, already it is there on the shared on the particular site. I think my all the documents are already uploaded over there. Okay, maybe the previous version, but almost the same thing is already available. So you can also check around those things. Okay, you will find the same points. You can go through with this one. Okay, these are important points here. Plus, I will share with you a glossary part. I'll just cross check. I hope that is also available. So you can, if it is available, you can check that glossary or the definition part and easily understand that thing. You can easily go through with that part. Please note down if you face any difficulty in any of the terms. Okay, try to complete that glossary part as soon as possible. Now, few things which I would like to tell you, so that you, when you start reading these terms, you can get it very easily, and these are very important terms. Adverse drug reaction, adverse event. What is the difference? Both are almost same thing. Adverse drug reaction. First, whatever happened after taking the drug. Okay, your blood pressure shoot up, or you started feeling stomach pain. Anything happened, that is actually called as adverse event. Okay, but when there is a causality relationship established, that means your doctors, when they start checking this case, and in the pharmacovigilance department, when they started checking the case and they started identifying that there is a some relationship is there between this event and this drug, then we can say that this is adverse drug reaction. If there is no relationship established or very minimum relationship is there, which is like neglected relationship, we will not consider it as an adverse drug reaction. We will just say it is an adverse event. Okay, so that is the entire thing. So any side effect in your mind right now, you can consider that as an adverse event. If the relationship between the drug and that side effect established, you can say it as an adverse drug reaction. There comes the blinding and masking term. So blinding, hope we are going to study in detail in the next session, next session, designing session part. For now, you can understand that when we are taking the drug into the clinical trial, okay, we are actually masking this drug. Masking in sense that even the doctor sometimes don't know. Sometimes it happens that the doctor knows, but the patient don't know that which drug he or she is getting, placebo or a particular drug. Placebo means a tablet without active ingredient. If this particular person is getting the placebo or active ingredient drug. Okay, so we are actually masking this either to the patient, or even sometimes it's like double masking, that is also doctor or coordinator, they also don't know what medication they are giving. It is as per the particular designing. Okay, whatever is designed the clinical trial, the doctor and the coordinators, they know that they have to give only this color or this medication to this particular group of patients. Why this is done? So that we can have the transparent results. Okay, there should be no biasism. Case report form, this is another thing. Okay, we'll also cover up that part. So basically this is for taking down day-to-day cases that you have given the drug, what is the result from that particular drug. So you are going to take it in the case report form. Informed consent, as I mentioned earlier, this is in the very beginning stage when you are asking the participants to come in the and participate in this trial. You are actually telling them, declaring everything what is going to be there in the particular clinical trial. That is when you need to come, what you need to get, okay, what is expected, how much monetary thing or insurance or other things you will get. Everything is written in clearly to them. It is similar like you are signing an affidavit. Okay, so you are signing an informed consent. Comparator means when you are comparing your drug with one of the marketing competitor or a placebo. So those with which you are comparing your drug, that is called as comparator. Okay, CRO, clinical research organization. So you have already, I think you learned about this already a lot, and hence you guys are actually here. You may want your career in CRO one or the other day. So basically, CRO is the place where we mostly do our this entire clinical research. Multiple in some big CROs, there are multiple clinical trial projects goes on. Okay, so that is the particular location where we are running these clinical trial projects. Essential documents, separate chapter again on the essential document available. So it depends that how much time we have, since this is entirely a short course. I'll try my best to cover up these things. So essential documents is basically all the documents, including the protocol, informed consent, and there are so many documents which are important as per the clinical trial. Those documents are called as essential documents. Okay, there are other documents which are actually not required for the clinical trial. Maybe they are just a supporting documents in doing the clinical trial. Those are not essential documents. Okay, we will see the entire list of essential documents too. Then there is a GCP, good clinical practice. So this we will do it in the after historical part. We will do the GCP, ICH GCP, and I will take you through the main GCP guidelines. Good clinical practice. Okay, so this we need to understand before we start doing the clinical trials. Okay, then there comes the vulnerable subjects. What is vulnerable subject? So vulnerable subjects means subjects which are very vulnerable to be misused, like children's, like illiterate people, like elderly people. These are vulnerable populations. You can mislead them and you can misuse them, especially for these vulnerable subjects. Okay, the authority asks to make sure that they understand at the very basic level about what is clinical trial, what is going to happen with them, what is going to happen with them. Okay, so that is another important thing which is important here in the informed consent term. Okay, sometimes it happens that if children are participants, so as per the informed consent, it is clearly mentioned that if children are there, then there should be some adults, okay, or some take carers. They also need to be there to sign off the consent form. Unexpected adverse event. Now there are some side effects which are expected. That means you know that this drug can cause, and this is happening in the patient. There are some side effects which are very new to you, okay, and that comes first time to you and not as per the document. So you have actually investigator's brochure in your hand. That is a document which a medical doctor in the clinical trial keep. That is called as investigator brochure. So if this side effect is mentioned in the investigator brochure, it is considered as expected. If it is not, it is considered as unexpected. Okay, and there is trial site. So trial site is there. Okay, trial site means our PROs, basically wherever we are doing the trial. Sponsors, who is putting money and doing all this thing. Okay, the main project owners, the companies, okay, pharma companies, who is pouring money and giving you the protocol and all the important material and other things which is required. Source document are the raw documents, okay, which is available with you. So these are not the essential documents. These are the documents from where you actually get the information and you create your essential documents. Those are called as source documents. When you go through the entire glossary, you can understand this, like, just go with the lines. You will understand more in better in the source document part. Okay. Protocol, as I mentioned, this is basically a document which we create in the very much beginning of the trial. Okay. To tell that everything, all this thing is available, which we need to complete. This is our design. This is how the auditing going to be done. This is how we are going to get the investigational new drug, okay, and how we are going to store them, how we are going to discard them after clinical trial. Everything is going to be written in a protocol. It's like a master book. Randomization, I'll try to answer in 1 or 2 lines. The description I will provide you in the another presentation in the designing, where we understand randomization, blinding, all those things. Randomization is basically when we are putting a particular person in a particular branch of our well-designed clinical trial. So in the clinical trial, we have, let's say, multiple arms are there. So you don't know you are in which arm. Maybe you are in the A arm and you are actually supposed to take placebo, not the active drug or not the other company comparator drug. Maybe you are in the arm where you need to take actual investigational new drug. So randomization is you or doctors and others have no idea there is a process of doing randomization by any randomly method you are going to be in one of the branch. So it is to keep the transparency. Monitoring is basically, as the term says, we are going to, there's a monitoring team which keeps on monitoring the particular entire way that how the clinical trial is conducted, if there is everything going as per the protocol or as per the GCP guidelines or not. Okay. Investigator is basically the doctor. Tomorrow we are going to understand the responsibilities of investigator, a very important part of the clinical trial, who is conducting the trial. IRB, IC body, I also park this particular part for tomorrow when we are going to understand in depth IRB, IC. Okay. Serious adverse reaction. Serious adverse reaction is basically the particular ADR or AE in which there is a serious outcome. That means with the AE, there can be hospitalization, there can be death, okay, there can be prolongation of hospitalization, there can be the birth defect, there can be physical disability. All this comes under the serious adverse reaction when the outcome are these. Regulatory authority, I hope all of you know, okay, mainly who is keeping an eye on everything. Investigator brochure, I have just mentioned a few minutes ago. So the big book which we keep during the pre-marketing time, the doctor keeps it, okay, and everybody can see if these events are expected, unexpected, and the other details of the product. So that is in the investigator brochure. Multi-center trial. Now, multi-center trial is multi-centric phase 3. If you remember, we have separate geographical regions in which we are running the multi-center trials in the different centers. So that is called as multi-center trial. It is actually phase 3. Investigational product is our product on which we are running this investigation. The drug candidate, it is called as investigational product during the phase 1, 2, and 3. And then compliance. Compliance means as per the time, are we compliant or not? So as per the as per the regulatory guidelines, if we are sending all the side effects information to the authority in timely manner, are we submitting all the reports required by the authority in timely manner or not? So that is how we are compliant with it or not. Okay. Placebo is, as I mentioned, any drug, any medication, or any formulation which we are creating, not medication, any formulation which we are creating in which there is no active ingredient available, only excipients, okay, they are available. So these are called as placebo. Okay, there is no active ingredient, only excipients, binders, disintegrating agents, all of these are available. Okay. So this is basically the most common used terms in the clinical trial. So you have to have an awareness of this. Apart from these terms, you will find another maybe 20, 30 more terms available in the glossary. Okay, I'll just go with that glossary part, and you can understand those terms from the glossary. If there is any challenges, please note down. Let me know tomorrow in the beginning of the session itself. I will try to explain that part also. Okay. So I think we are almost at the end. Yeah. Any question anybody have? I'm open for the questions if there is any. No questions? Okay, fine. So let me see 1 second. I'm sorry, wait. Okay, Dinyashwar, sorry, what is the question? Okay, I'm waiting, no issues. I'm not closing the session. You can ask me. I'm just checking the another piece of information. Do you want me to open the slide again, Mr. Solanki? Oh, Ms. Solanki, Dinashwar Solanki? It is not fixed. It is not fixed. I have said that yes, approximately 10 to 15 years required. Yes, but it is not fixed. It can be early, it can be late. As I have mentioned earlier, if you have attentively listened to me, I have mentioned that COVID vaccines and other things are not following the regular clinical trial protocol or timeline. These are expedited clinical trials because of the conditions which is there right now in front of us, and there is actually no other medication or vaccines available to handle this thing. For this, there are some exceptional conditions are available. Okay, in our books, in our guidelines, they are following that exceptional conditions. Okay, so hence the COVID part, please consider this as an exceptional thing. This comes once in a while in a lifetime. So if that is there, then definitely every country, every authority wants some kind of drugs as soon as possible. Yeah, of course, scared of this thing because this is for one of the deadly virus thing, right? So nobody wants to take chance. Great. Okay, so I will take you quickly to one of the another slide so that you can read that part also. It's a very small thing. Okay. So drug classification. So whoever is from the pharma background, medical, I hope you all know about the drug classification much better even than me. But still, try to go through that part before your interviews and before other things, because you may be asked about one or two other drugs too. Okay, so you may be you should be answerable, or they may ask you about your favorite organ system or something like that. Okay, so you need to answer, let's say cardiac, so they will ask you asking the cardiac drug. So please be ready with that part. This is a classification I'm taking you is a WHO ATC. Okay, WHO anatomical therapeutic classification. As per that anatomic therapeutic classification WHO, these are the main categories. V, you can see almost I think 14 categories are there in which you can keep all your drugs. So they are actually divided in this pattern, that is alimentary and tract and metabolism, blood and blood forming organs, cardiovascular system, dermatologicals related to skins, genitourinary system, and the sex hormones. Those drugs you will find in this category. Systemic hormonal preparations, excluding the sex hormones, which is there in the G category, and insulins. Okay, so these are the other drugs which we need to understand. Then there comes the anti-infective for systemic use. Okay, so if we are using any those products, you will find those drugs over here in this category. Anti-neoplastic and immunomodulating agents, musculoskeletal systems, nervous systems, antiparasitic products, insecticides and repellents, respiratory system, sensory organs, and various. So these are the main categories as per the WHO ATC classification. Yes, there are various classifications available even as per our pharmacological books, which we have pharmacology books. So you can understand any classification, whatever is best for you. Okay, or you can go with this classification, which is internationally available as per WHO. Any classification you can understand, not an issue. The main thing is you have to at least understand and remember few class of the drugs. That is important. Okay. There is one particular link I have provided over here. You can go directly on this link. Okay, you can type it around. This is related to one of the Canadian website, and on the same basis of this classification, they have actually classified all the medications. Okay, in the various sub levels, these are available as per WHO ATC category. There are actually 4 or 5 labels. Okay, like first there is a like a therapeutic agent level, pharmacological level, as the medical chemistry level is there, and hence slowly they are categorizing all the products, and at the end last level you will find the product name. Okay, let's also see that part. Allow me 1 second if I can take you over there. So this is the screen I'm actually talking to you. You can see all these categories are available. You can open any category, let's say cardiovascular. So you can see at the first level, all these pharmacological levels are there, with beta blocking agent, calcium channel blockers. Okay, once you open any of this, you will move to the next level, like the therapeutic or like, as a chemistry level. You can see calcium channel blocker and diuretics. Okay, and then there is non-selective calcium channel blockers. As further you can go down and you will find one more level. You go down, you will find the drugs. Okay, amlodipine and diuretics, example amlodipine, and over here nifedipine. So there are multiple levels available, and under that you will find the specific drug available. This is as per the ATC classification. Okay, you can also anytime type WHO ATC and you will come to the this one also. I'm just showing you that anytime you would like to hang around this thing to understand, you can easily get it. So you just write metformin. You have so many metformins. Okay, it is actually categorized and given a particular term. Okay, so whenever we are using this term, there is going to be behind the bar, there is going to be some coding number. This is in the dictionary when we are feeding in the information during the softwares in clinical trials. So this is just to tell you, all in all, not to see much about ATC, just to remember some classifications. Okay, I'll share one of the document if I have right now handy. It is actually already uploaded in the clinical trial this session. You can also check this document. Let me see. Yeah, that PDF classification of drugs I have. Okay. So you can see this classification of drugs. This is also available over there. So hence you have to just go through with this classification of the drugs. Okay. And let's see cardiovascular. Okay. So if you are interested in one of the cardiovascular category, you can just see that there are further subcategories are available like renin, angiotensin, aldosterone system, beta blockers, calcium channel blockers. So this particular document will give you a handy tool to note down some of the good products in your mind when you're preparing for the interview. Okay, you don't need to memorize everything. You just need to focus on few categories and few subcategories so that you can answer right away. Okay, with every single drug, let's say over here it says lisinopril, there is a bracket and something is written, right? In the bracket, it is actually the brand name of this product. Okay, but I will suggest don't go with this brand name. Okay, you have to just remember the active ingredient or the generic name lisinopril, enalapril, ramipril, like that for that particular. So just 2 or 3 examples for that subcategory. There are so many subcategories are there in cardiovascular, in respiratory there are 3 subcategories. You have to just select some main category, some subcategories, remember 1 or 2 examples of each subcategory. Hence, if any question comes up related to your favorite organ system, you can select 1 or 2, and if any question comes related to further subcategories or examples, you can provide right away. That's the whole purpose to have these documents. Apart from that, nobody will go much in depth to understand the mechanism of action and other things. Okay. Being a medical and in administrative sector, what can be the application of clinical research? See, clinical research itself has its own importance of identifying or getting some new drugs, if it is a treatment or preventive type of research. Others also research shows that if you do observational type of studies, you will come with some data which you will present. Okay, medico, of course, being a medico, like you are very good or you can easily go with the particular clinical research if you have interest. You are completely eligible for that. Administrative sector, frankly speaking, in the particular clinical research, if you are from the administrative background, Tamshwita, you can more also go in the project management type of department in the clinical research. Okay, and there are few other departments in which you don't need to work directly on the branch level. Okay, you may work in the as a administrative roles where, like, you are checking the documents and other things. But if you are interested only there, then only. If you are interested more in laboratory data, then you don't need to go for administrative type of task. You can go in the laboratory as a coordinators, monitors, and then you can work over there. Okay. So guys, I'll take a pause here. I know that you all have some reservations. Okay. And I tried my best to provide the knowledge, whatever I have over here in first session. Please go through this entire thing. I hope that this is already uploaded, okay, in our session. If you all have the access to the portal, you can easily have that, all the documents available over there. I hope some of you already completed that part too. But just go through again with these things what we have completed today. Okay. Let me know whatever the questions tomorrow. So we'll start sharp at 9:30 tomorrow so that we can cover it up in timely manner. All right. Take care, all of you. I'll see you tomorrow. You all have a good day.